the formation of nervous system. Now, what is going to happen? So, what we have so far, we are saying we have the three layers. Outer layer, then you will have a inner layer, and then you have these three layers of the cells we have, right? So we are saying the cells which are right here in the outer layer, these cells are ectoderm cells. Right? And we are also saying that then right here, the mesoderm which is here, this mesoderm, it is special type of mesoderm because this mesoderm is your notochord, right? So this is your notochord. And if you look like this, so this is kind of a cross section. But during this process, what happened after the gastrula is formed, we are not kind of a circle, right? So the embryo is elongated like this, right? So in this way, if I'm trying to draw it like this, I will say, okay, looking from the top, I will say this ectoderm is there, and this ectoderm, draw the same color. So for example, this is the ectoderm right here, okay? This ectoderm is underlined by here, the mesoderm, and this mesoderm, which is like the ventral side, right? So if you look here, so you should have here your precaudal plate, right? Which is your head mesoderm. And then right here should be your notochord, or coda mesoderm. So this all will be your coda mesoderm or notochord, right? So we are saying that the area above this, so this is that area above this, the notochord and head mesoderm total, I'm just saying that because this is elongated like this, right? So you cut cross section anywhere, if you cut cross section here, then this will be the head mesoderm here, you cut here, it will be notochord here. Making sense? So what is going to happen, this notochord, I'm just writing here notochord, right? So this notochord, what it is going to do, it is going to tell this ectoderm cell which are above this. So notochord is going to induce the formation of neural plate. Formation of neural plate. And that neural plate will happen in the ectoderm cells above the notochord. So formation of neural tube is going to, neural plate is going to take place. Now, so this neural plate, what it's going to do, is going to involute. This is going to involute, and this is going to involute into the body. Because nervous system is not outside, it's inside the body, right? So this need to the neural plate which is formed, that is involute inside the body, and what is going to give you is going to form a neural tube. And that neural tube will be a precursor of brain and spinal So the whole game is about forming tube, one tube, next tube, like that. Okay, so far it is okay. Then, so the part which is here, above the node of that is going to give you the neural plate, right? Now the other part which is away from this, so I'm saying away from this, or I should do it like this. Other part that is away from this. So look here, I'm leaving a gap here, right? So other part that is away from it, that other part of ectoderm will become epidermis. Okay? Now, this epidermis is the largest organ of the vertebrate body. Forms a barrier and that barrier is from outside in the body. So that barrier outside in the body is essentially your epidermis. Now, then here is this portion of the cells. Now we are saying this part which is above is becoming a neural plate, right? And then we are saying the part of this epidermis or ectoderm layer which is right here on both sides. Between the part of the ectoderm that is going to give epidermis and this part is going to give the neural plate. This part which is here. This part is going to become neural crest. Neural crest. And this part is going to give you peripheral nervous system. Peripheral nervous system and melanocytes. And other thing also I'm going to talk, just a kind of outline here. And melanocytes, like the pigment, the color pigment of the body. Okay? Now, so here is what is happening. So if we go a little bit bigger about this, like what is happening here, right? We'll keep this diagram on and we'll find out what are the formed. So if you look here, if you remember why we are, any doubt here? Mason, you're okay?
No, yeah, I'm good now. Yeah.
Okay. So if you look here, what we are saying that this part of the ectoderm that is away from this mesoderm, which is coda mesoderm and the notochord, right? What these guys are doing, we said they induce the ectoderm above them by inhibiting BMP and Wnt signaling, right? We said if you have both BMP and Wnt signaling inhibition, you get brain. If you get only Wnt signaling inhibition, you get the spinal cord, right? So, part here does not have any BMP. There is no BMP. Oh, sorry, there is no inhibition of the BMP. So this part, which has very high BMP, so that part is going to become what you call surface ectoderm. Very high BMP, surface ectoderm. So what is the component of surface ectoderm? Number one is your epidermis, which is essentially your covering of the body. Then what you get is your hairs coming from there. You get nails also from there. You get your sebaceous glands sweat gland. From there, then you get your olfactory epithelium. From there, you get dental enamel. From there, you get your cheek epithelium from there. So all these things, and then your eye lens is also from there. Your cornea is also from there. And then part of the pituitary gland, the anterior pituitary is also from there. So all these things are coming from this area right there. Okay? Now, when we are going a little bit further, we are saying let's come here. Okay? Now think about these cells which are the mesoderm, the coda mesoderm, the notochord, all those guys. These guys are secreting BMP inhibitors, right? So if they are secreting BMP inhibitor right here, so say, let me draw it in a different, say they are secreting BMP inhibitor, they are saying BMP inhibitor. This BMP inhibitor is secreted by them going directly up, right? Now some of these BMP inhibitor, they might leach out on this side. That is possible, right? That's what happened. So in this area where it's direct there is no BMP. But here in this area, you can have moderate amount of BMP. Okay, and when you have the moderate amount of BMP, you become neural crest. And neural crest gave you multiple things. It gave you enteric nervous system. Enteric nervous system is the nervous system of the gut that controls the gut motility, gut absorption, gut secretion, and we also call it second brain. And now, so I don't know, you will never study this in this curriculum about that. So enteric nervous system is also, this I want to give 2 minutes on this. Enteric nervous system is also called the second brain, right? Because it can control all the gut activities without the influence of the central nervous system. So think about when you feel hungry. Your gut needs to prepare before you eat food. So you need to start salivation, right? And then your gut starts secreting all the enzymes so that they can digest the food. That should happen before that. And then when your food is digested, that food should be absorbed by the gut so that it can reach at the right place. That signal also comes from the enteric nervous system. And then when you eat the food, we all know that this disease called diabetes, right? You have a lot of blood sugar. So what sent that signal to the pancreas? That signal is sent by the enteric nervous system to pancreas to start making the insulin, that the food is there. Then when you're full, there should be some signal that is telling you that you're full. That signal is also controlled by the gut. Now this is the gut motility, how the food travels down the gut. Now this is one thing. Now throughout this time, I'm telling you always that we are a tube, right? Think about, we are tubes and there are many tubes inside those tubes will form the neural tube, all those things, multiple tubes in the body. So think about, we said that the skin is or epidermis is the largest organ that covers the body, no interaction from outside to inside. Think about that, all the neurological disorder. This is kind of interesting thing. So you can have a mutation in, for example, amyloid gene, although mutation have little link with neurological disorder, but mutation, they do not explain all the neurological disorder. For example, Parkinson, only 3% have mutation people. Other people don't have any mutation. Alzheimer, 1 to 2% people have mutation, other people don't have mutation. Huntington, same way with every neurological disorder. But here is one thing, think about those mutations which are, so what is happening in Parkinson? Parkinson have a mutation in gene called Parkin1. Parkin1 is an important gene involved in somehow regulate the electron transport in the electron transport chain of the mitochondria, ATP production problem. Think about a gene which have problem in the ATP production. Why that gene cause only problem in the nervous system? And every cell need ATP, right? Your cardiac cell need ATP every time. Then why not all the cells of nervous system die? Why a specific set of the neuron die? For example, in Parkinson's, substantia nigra neuron, which are right here on the back, they die. So what is there? So now think about, now people are talking more about, it's not genetic, we call them, so when you don't have any cause, you call them idiopathic. So most of the neurological disorders are idiopathic. Now they are finding whatever we eat, whatever we are exposed to, that is the problem. Now think about that Parkinson disease is a central nervous system disorder. Central nervous system means brain and spinal cord. Peripheral nervous system, but what happened in Parkinson, you got stuck like this, shaking. That's the motor neuron are sending signal here to the skeletal muscle. But what happens, Alzheimer, you forget everything in the brain. But now what they are finding, for example in Parkinson, before you see anything like this, those people are severely constipated. And they also lose the smell. Now that's what I'm telling you too. Think about that our nervous system is totally protected by skull and vertebrae central system. But what about, then what is the exposure route of the central nervous system? Two route. You smell. When you smell, right here are the neuron receptors. They can be taking whatever toxin you're smelling to the central nervous system. Second is your food. When you eat the food, in the gut you have enteric nervous system. That enteric nervous system directly connects to the central nervous system. Now people have found, like even the 10 years before, I was telling the constipation, then you look at the neurons, they find all those classical symptoms of degeneration are present in the enteric nervous system before they are present right there. That's what we are doing in the lab. We are trying to find out the early marker which are in the nervous system, so you can predict Parkinson is going to happen 10 years before today. So same with the diabetes, most of diabetic patients, they are constipated. People who take drugs, they are all constipated. People who drink a lot of alcohol, you have a lot of gastric problem there, right? So this nervous system is critical, what we call enteric nervous system. Now this nervous system, just to give you the background, what that is. So that is a critical nervous system. It's not like just in the gut and controlling that. So that is coming from the neurocrest stem cells. Now, the other thing which is coming from these neurocrest is peripheral nervous system. Peripheral nervous system. Now you'll say, what is peripheral nervous system? Peripheral nervous system is all the nervous system that give you, you touch here, all the sensation. You know, this year's Nobel Prize was for those two guys who said, who received the Nobel Prize. They discovered the receptor for pressure and all those things. So all the pain actually which is coming from here to here, that is all through these sensory neurons, what we call sensory nervous system. So peripheral nervous system, that is coming from the neurocrest. Then your sympathetic and parasympathetic. Parasympathetic nervous system. That all is coming from the neurocrest. Then melanocytes. Those are the color pigment. They are coming from these neurocrest cells. And then facial cartilage. Facial cartilage, that is coming from there. Then your adrenal medulla. So there is an adrenal gland right above the kidney, and that gland is called adrenal gland. So you cut that gland. This is also important. You should know this. So on the kidney, so this is for example kidney, right? There is a gland above that is called adrenal gland. If you cut the kidney, this gland, so it has internal portion is called adrenal medulla, outer is called adrenal cortex. This adrenal medulla cell are making a lot of neurotransmitter called dopamine neurotransmitter. And the neuron which die in the Parkinson are all dopaminergic neurons which make dopamine. So people were trying that you can take this adrenal medulla, these cells, and inject those cells in the brain. And there was some clinical trial done, some successful, some not successful. So this is coming from there. And then you have that, we said enamel of the teeth is coming from here, right? Then in the teeth, in the tooth, there is called dentine, right? So that dentine is coming from here. So all those things are coming here. Now, this is moderate BMP. You can see that just the levels of BMPs are doing all the trick, right? Now, if you have this area, the area what we call neural plate. In different colors so that there's distinction there. So now you have the neural plate. Now the neural plate will have no BMP, and they will have high SOX enzyme. SOX 1, 2, 3, these are three. Transcription factor, I'm not saying enzyme, they're transcription factor. So they have no BMP and high SOX there. And what they're going to do, they're going to make brain, they are going to make spinal cord, and what is in the eye which make you see? What you call that portion? Retina. So eye retina is coming from here. Now the cornea is coming from here, from the other. Now you can see that this is kind of, if you sit back and think that how your eye is formed. You know, you have eye lens, you have eye cornea, you have eye retina, right? So cornea and lens are coming from this area, and this area is telling you that, hey, you are making the retina. How those cells move is kind of like fascinating. So these are like different things which are coming from the ectodermal area, right? And how they form, we are going to talk. So just go over it, and then we'll talk how the neural plate is formed.
The neural plate includes inside the body?
Yes, the neural plate includes inside the body to form the neural tube. Which is the precursor for the cord.
So we have like 3 layers. The neural cord above that, ectodermal that becomes the neural plate. Neural crest are to the side of that. Neural cord induces formation of neural plate and ectodermal cells above it. Move inside the body to form neural tube and it's like precursor to the neural cord. Other parts of the ectodermal become surface ectodermal did all of that stuff. Moderate BMP's neural pressure, low BMP has SOX 1, 2, and 3 neural.
Everything is okay? Okay, now. This process, now, we are going to talk about what we are saying is neurulation. Neurulation. Now, what is neurulation? Neurulation is process by which three ectodermal areas are formed. So this process is called neurulation, okay? And those areas, we just talk about that. And the embryo undergoing neurulation is called neurula. So embryo undergoing neurulation is called neurula. Okay? Now, so what is there? How this process is happening? So we are saying that, so we have this outer layer. Say this outer layer, this is your neural plate. Right? And this whole thing is your ectoderm. And beneath, right here, is your area, what you call so this is your mesoderm before, under that, right? And right here is your notochord. Okay? And beneath the notochord, what you have there is you have this structure right under that, and this is your endoderm, and this is your archenteome. Okay. Now, this neural plate is induced by the notochord, right? So neural plate is characterized by high SOX1, SOX2, and SOX3 genes. Now, these SOX1, 1, 2, 3, these are actually transcription factors. These are transcriptional factors. And what they're going to do, they're going to induce the neuronal genes. That's what their function is. They want to induce the neural gene to form the neural tube. And then they will inhibit BMP to inhibit the formation of epidermis and neural crest. That's what they're going to do. Now, so what is going to happen? So you're going to have, this thing should get in, okay? And that is happening because this structure actually invaginate like this. This is your neural plate structure. This invaginates like this. And then when that is happening, this is your neural cord structure. That structure is also kind of changing its shape like this. And in the center, you're going to have your rest. All of this is your archenteron. This is your endoderm, like this. And this cavity which is now formed there, this will be now called gut cavity. Remember, it's not only, there's so many things going on, but we are just talking about how this tube is being formed, right? And right here, this structure which is formed there, this is called neural fold. This structure is called neural fold, okay? And then ultimately what is going to happen, this is going to lead to a situation when you'll have this whole neural tube coming inside like this, and this will be surrounded by epidermis, and then you will have your notochord still there, and then you will have your this will be all your endoderm, and this will be your gut cavity. And this thing is now your neural tube. A tube is formed in a tube. Okay, now you have two tubes here, gut tube and the tube of the neural tube. Okay, now how this thing is formed? This is a very simply oversimplified thing to show you that, okay, this is going to invaginate, make a neural fold, and then this tube will close and you will get a neural tube inside. So this process we said is called neurulation, but how this process actually occurs, how this thing happens, okay? So we're going to talk about that. So neurulation process, it yeah.
What starts the neural fold, like, to take place?
Say it again.
What causes the neural fold to start?
We're going to start. Yeah, that was just the overview thing, right? Then the fold takes place, neural fold occurs, and then form a tube, and then you get that. Now this process of neurulation is actually a two-step process. Number one we call it primary neurulation, and the second step is called secondary neurulation. And this secondary neurulation takes place only in the tail region. So for the spinal cord and brain formation, this process is not involved. So primary neurulation is mainly involved in the formation of spinal cord and the brain, okay? So now what happened? It has multiple steps there. So the primary neurulation, it mainly occurs in 4 steps. Okay, now what is happening? The first step is what we call it extension and folding of neural tube, and that's what we are getting your answer right here. So what is happening there, that again, if you look at this diagram, so we are saying right here is your neural plate that is formed there, right? But this neural plate, say in the beginning this embryo was like this, but now this embryo is kind of extending like this, right? Head tail is being formed, extension like this. So it means initially if it was like this, now it is becoming like this. So this neural plate, which is supposed to be on this area, should continuously divide. Making sense? Or no?
Not really. Can you repeat it one more
Let me explain. Initially, your cell is like this. We are round cell, but we are no more round structure, right? So what we are like this. This is our mouth. This is our belly. We don't have tail, but mouse can have tail, right? And 4 legs, hind leg. We are like this. So if this was the structure in the beginning, we had the neural tube here, or neural plate here, right? So it means if we are this, now we are becoming like this. Making sense? So if you are becoming like this, so this neural tube, neural plate which was here, this should divide, these cells should divide like this, and also extend. Making sense? Now, so they should, and now think about, if this is the body right here, so this is now, think about, now this is the whole neural plate. Let me draw a different color. Now this has become the neural plate right here, right? So this whole neural plate should fold, right? Only then you will have the spinal cord and brain. If you think like this is your brain right here and this is your spinal cord, so this whole thing should come inside and fold. Now, so the first step is what we are saying, extension and folding of the neural tube. So what happened? Extension. This is extending from here to the whole body. So that means what happened, the division of the neural plate cells. And when they are dividing, what is happening, that division, so think about, if this division is taking place, if you look at our vertebral column, that is in the center. We don't have the spinal cord covering all the back, right? So it means that division which is going to take place, that should not be breadthwise. Should not be like this, right? So that division should be like this. So division of the cells, and that is happening preferentially, and preferentially only in one direction. That means across head to tail. Right? Making sense so far? Should be like this. So it is spreading across head to tail. So think about now what happened. Now think about that has spread from here to here. Now we want to talk about how it folds. Okay? So think about like we have, so this is your neural plate. And this neural plate is surrounded by what we said here will be your neural crest cells, right? And those neural crest cells will be surrounded by the epidermis. Say right here is your epidermis. So will be all your epidermis. Okay. So now what happened, that right here is your notochord. Notochord. Okay. So what is going to happen? Now we have talked about extension, we are going to talk about the folding, how the folding is. So this notochord is right here. So I'm drawing a little bit gap there here. A little bit gap there. Now notochord is going to start secreting the factors. Okay, those factors we'll talk about what are those in a minute. But it starts secreting the factor. And what is this? These are all cells, right? These are all cells. And the cell above the notochord, they actually come and they attach to the notochord. Let me show you what is happening here. So this is the next step. So I'm just drawing this area of the neural plate. And these are the notochord right here. What notochord does, it induces the cell above them to shorten. First they shorten, okay? Cell above them to shorten, and then think about this. They're like this, and right here is your neurocrest, and above after this is epidermis. Now think about it. They are shortened here, and then what it does another thing. First shorten them, attract them, and then change shape. And what that shape changes. These cells are now like this, shortened, but they're like this, okay? And what it does, it tells them to change shape, that from here, there, you see this and this length is same, right? It changes them in a way that they, instead of like this, they become like this. Okay, so changes their shape. So the cell change shape. First step, attract them, shorten them, and then change their shape. Now think what is going to happen. If these cells after this were like this, they were like this, right? But now these cells have changed the shape. Let me draw them here. Now these cells have changed their shape. They have become like this. They have become like this. So what is going to happen? These cells which were now like this, they are actually going to become like this. Making sense? So this is the next step. And now right here is your notochord is there. Notochord is here and these cells are there. Now, these cells which are, now they're making a hinge, right? It's a hinge is formed there. So these cells are called medial hinge point cells. So far it is okay? Now that is how the fold is formed. That makes sense? Okay. Then what happened? Then we have more cells here. So these were your neurocrest cell. These were neurocrest cell. And then those were, so there are many cells like this. And then you have your epithelial cell and these nucleus cells. So now what happened? Now the next step happened, what we see is convergence. Now the cells which are here, now I'm just drawing 3-4 cells, but there are many cells, right? So what happened? Now the cells which are here on this side, right here, these cells, they change shape, like the previous cell had changed the shape. Okay? So these cells, they change the shape. And that is the step 3, we are going to talk about convergence. Okay? So what is going to happen? Now, if I'm drawing it like this, I'm saying, okay, let me draw this. So we had these cells right here, we had these cells right here, there was your notochord, and what had happened, this has caused these cells to be like this, those cells were like this. So what happened, now these cells change the shape there. So they also become the cells which are like this. Okay? So now if these cells have changed shape, what is going to happen? These cells are going to come like this. the green cells will be there, like this. And to those, so for example, this is still the green cell. This is the green cell, which is your nucleus cell, to which these red cells are attached, but then you have more and more red cells on this side. So far it is making sense? Now this hinge which is formed there, this is called dorsolateral hinge point. This is called dorsolateral hinge point. Okay? Now what happened? The next step, step 4, is called closure. And what happens in this step? That you get all these cells, which are your epidermal cell right here. And then what you get here, right here, you get these cells, they have formed a tube like this structure. And here are still, you have those green cells there, nucleus cells. Okay? And then what happened? These nucleus cells, they move out from here and they go away. And what you get ultimately, a structure where you have this epithelial cell or epidermal cell, and you have a neural tube formed here, and all the green cells, they are moving to the different parts of the body where they were supposed to go. And you still have your notochord here. Now you will say me that, what is happening here? So far I have explained you the mechanism. I said, hey, these cells change the shape, right? They change the shape. Something happening from the notochord, how that happened, I'm going to talk in the next topic. But what is happening here? What caused these cells to fuse, right? We are saying that these cells, the hinge is formed. Okay, the hinge is formed, that's fine. But why the hinge formation caused these epidermal cell, ectodermal cell, which are now epidermal cell, to fuse, right? What caused these cells to fuse, your neuroplate cell, and what caused these neural crest cells to go away? Okay? So what is happening there? In the beginning, all of these cells, they all express EP-cadherin. EP-cadherin proteins. So they all have EP-cadherin proteins in those cells. So they are all attached to each other. Making sense? When this process is occurring, this is occurring, still they all have EP-cadherin. When this convergence takes place at this time, what happened? The cells which are the part of the neural plate, these cells, they start expressing, and when I'm saying expressing, I'm not talking about gene expression, I'm talking the protein expression, right? So they start having the protein. They start expressing neural cadherin, also called N-cadherin. Now remember, cadherin are joining protein, join cell to cell, right? So now, okay, so good. These have started expressing N-cadherin. These are still having EP-cadherin, still EP-cadherin. But what happened? The cells of the neural crest, which were initially expressing E-cadherin, they actually stopped expressing any cadherin. they stop expressing cadherin. Now think about, these cells stop expressing cadherin, so they will no longer stay there because they don't have cell adhesion protein, right? So this cell will move away. And this cell, they are N-cadherin, they are saying, I'm expressing N-cadherin, okay, then join to each other. And this cell, they are still expressing EP-cadherin, okay, if they are gone, that's okay, let them go away, we'll join with each other. So they make EP-cadherin. Making sense? So what we are saying? We are saying, this process of neurulation, the process ultimately is you want to bring neural plate inside the embryo. How to bring that inside the embryo? We said when this process is taking place, we call it neurulation, and embryo undergoing this process is called neurula. We are saying this is a multi-step process. Basically two major steps, primary neurulation and secondary neurulation. We didn't talk about secondary neurulation, we focus our attention on primary neurulation. We talk about secondary neurulation. We are saying primary neurulation can be divided essentially in 4 steps. The first step here is extension, which is division of the neural plate cell. And then when that division takes place, that neural plate extends from cranial to caudal area. These cells grow. And then we are saying there is folding is going to take place. And that folding is going to take place because the notochord is there. Notochord is going to stimulate cell above them to shorten their size and attach to them. And then notochord is going to change their shape to wedge-shaped cell. They become wedge-shaped cell, and that create a hinge there. And these cells which have become wedge-shaped, these are called medial hinge point cells. That cause the elevation, this folding take place, right? And when that folding take place, that elevation has taken place, the cells here, they also change their shape and they become wedge-shaped cell, and these cells have become wedge-shaped cell. And this is called dorsolateral hinge point. And what that does, that brings the cells which were surrounding them to close together. Now during this process, what we are saying, what is happening, before that, every cell was secreting, making EP-cadherin joining them. But when this dorsal hinge point is formed, this structure is formed, the cells which were neural plate, they start expressing N-cadherin or neural cadherin. The cells which are here, which are neurocrest cells, they stop expressing any cadherin. And the cells which were epidermal cells, they continue to express EP-cadherin. Now because these cells are not expressing any cadherin, these cells leave their space. When that leave their space, so this space is now empty. If there is no space there, these cells are secreting EP-cadherin. They will join to each other. They are secreting N-cadherin and they will join to each other. And ultimately that leads to formation of the neural tube, and the neurocrest cell, they go away from there. Making sense? Any doubt here? Okay, talk to each other for a minute, then we'll go further.
These are like 4 steps. Would you say it's the extension, folding, and routing, then the cells change shape, then convergence, and then closure? That's what I did you sort of think about correlation? Classical there's 3 ectodermal areas are formed, which wasn't all of it. And then the embryo undergoes neurulation is called a morula. The neural plate has isocs 1, 2, and 3, transcription factors, and they enhance the neuronal genes and inhibit BMPs, inhibit formation of the neural crest. Cell structure imaginates in Q2, and this is done by primary neurulation. Which starts with the cells extending and folding. Hopefully only go one direction at the tail.
What is the question?
So you said that the
And the cords are secreting factors and then the cell above them
So
attach to them.
this
And
is
shorten,
convergence, right?
they then change
Yeah,
shape
convergence.
and
And the
attach
closer
arms
is the fusing. So this is going
because
up,
they form a wedge shape.
and they fuse,
And
which
it's
is
called
going
the
to
medial
face the
hinge
closer.
point.
and then
Convergence
do you
happens
have an idea for the first
because the
two
cells
steps?
above
Yeah, it was
change
extension
shape
and folding.
form
It was
like a circle, then that's the dorsal lateral hinge point, and then closure, neural crest cells leave, and there's just
Any other doubt?
neural plate epidermis. And that's because cells at the beginning have all cells have E coherent, and then once they start converging, epidermis only has E coherent, and then neural crest cells stop expressing EP coherent, and neural plate cells start expressing neural coherent. So the cells with no EP coherent move away.
Any doubt?
Do we need to know the factors that the notochord is secreting?
We are going to talk about those. Yeah, that's our next topic. Whatever your question you're asking, we're saying we're going to talk about it. So, that's interesting. So you're asking good questions. That's good. How much time we have?
2 minutes.
Oh, then we don't have time. So we'll meet on Wednesday for exam, and then in the lab we'll start the process. Right? And when you're doing it, so you can even bring your drive if you have, so you can actually start copying the pictures at that time, right? So tomorrow we are not meeting, right?
Yeah.